Welcome to the deep dive. Today, we're tackling something that honestly sounds a bit like science fiction, reversing the biological age of your cells. Not just slowing aging down, but actually making cells act like they did maybe 30 years ago.
Yeah, it's a pretty mind-bending concept when you first hear it.
So our mission for you today is to map out the science behind this. There's this claim that changing just like 4 specific things in a cell could rewind that biological clock. Decades, apparently.
It sounds incredible, doesn't it?
It really does. We need to unpack how this could possibly work and, what it actually means for your health, not just adding years to your life.
Right. And to really get started, you have to understand how the sources frame aging itself. It's not just simple wear and tear. It's more like a progressive failure of communication within the cell. The signals get messed up.
Communication failure. Okay.
Yeah. The pathways that keep things running smoothly, that maintain the cell's identity, they start to break down. So we're focused here on boosting energy, making things work better, not just, tacking on extra years of poor health.
Right, functionality. I bet a lot of people listening are sort of doing the math right now, hoping their biological age is maybe kinder than their chronological one.
Probably.
And that leads us straight into this core idea. Your genes aren't necessarily your destiny, are they? You have the DNA, sure, but something else controls which parts get used?
That's absolutely central. It's all about the epigenome. Epigenome.
The epigenome. Okay, what exactly is that?
Think of it as the system above the genes. Your DNA is like the entire library of instructions, the books on the shelves.
Okay.
The epigenome is like the librarian deciding which books get pulled down, which chapters get read, and which ones stay locked away. It controls expression.
Ah, so it's the controller.
Precisely.
Yeah.
And when that controller, that librarian, gets, let's say, sloppy or damaged maybe through environmental factors, lifestyle, just time, the wrong instructions get read. That's basically what drives cellular aging. The cell starts expressing genes it shouldn't, or silencing ones it needs. It's noise in the system.
Okay, epigenetic noise. And researchers can actually measure this noise now. How do they pin down a cell's biological age, separate from just, years lived?
Yeah, we have some solid metrics now, 3 main ones, really. First, as you'd expect, is just looking at the overall pattern of gene expression. What genes are active, which are quiet. That pattern changes dramatically with age.
Makes sense. The cell's playlist, basically.
Exactly. Second, we look at telomeres. You've probably heard of these.
The caps on the ends of DNA, right?
That's them, like the little plastic tips on your shoelaces. They protect the important DNA underneath.
Okay.
Every time a cell divides, those telomeres get a tiny bit shorter. Eventually, they get so short, the protective cap is gone.
And then the DNA itself is vulnerable.
Right. It gets damaged, and the cell usually enters senescence.
Mm-hmm.
It stops dividing, stops doing its job properly. Think of a muscle cell that can't contract well anymore, or a skin cell making less collagen.
Got it. So gene expression, telomere length. What's the third?
The third one is maybe the most critical, mitochondrial dysfunction.
Ah, the power plant.
The power plants of the cell, exactly. They generate something like 90% of our energy. When they start to fail, become inefficient.
Energy levels just plummet.
Precisely. And that energy crisis is a huge driver behind so many signs of aging fatigue, cognitive issues, muscle weakness, you name it. It all ties back to energy.
So, okay. Gene expression, telomere length, mitochondrial health. Those are the big 3 measurements for biological age.
Those are the tools, yeah.
Which means the goal becomes figuring out how to tweak those factors, right? How to actually improve them.
That's the objective.
Now, before we jump into the really famous Yamanaka factors, wasn't there a key moment earlier, like in France, where scientists first showed this reversal was even possible in human cells?
Yes, absolutely. That was back in 2011. Really pivotal work. They took fibroblasts, common connective tissue cells from very elderly but healthy people, centenarians, over 100 years old.
Wow. Okay. Cells that were biologically very old.
Extremely old.
Yeah.
And they applied specific genetic factors to them.
And what happened?
Well, it was pretty astonishing. The cells didn't just, get a little bit younger. They completely reset.
Reset how?
They turned back into pluripotential stem cells.
Huh.
Essentially, they became like embryonic cells. Again, a blank slate, capable of becoming almost any cell type.
Whoa. So the old cell forgot it was a fibroblast.
Completely. And get this. Their telomeres were longer, their mitochondria were healthy and buzzing with energy, and their gene expression patterns looked like those of a very young cell, like cells from someone still developing.
That's almost hard to believe. It sounds incredibly complex to achieve.
It does, right?
Yeah.
But then came the Japanese research, the really big breakthrough in 2006.
The one that won the Nobel Prize.
That's the one. Shinya Yamanaka and his team. They showed you don't need some complicated cocktail. You only need 4 specific genes. Just 4.
Only 4 factors to do that total reset.
Just 4. We now call them the Yamanaka factors, obviously.
What are they? Can we name them?
Sure. They're OCT4, SOX2, KLF4, and c-Myc. These aren't just any genes. They're master regulators, transcription factors.
Meaning they control lots of other genes.
Exactly. They basically rewire the cell's epigenome. They tell the cell to shut down the old age program and reactivate the youthful development program. They fix that broken communication we talked about earlier.
Okay, that's incredible for cells in a petri dish. But then comes the big hurdle, right? Doing this in vivo inside a living person.
Right.
Because if you take, say, an old heart muscle cell and hit the full reset button with those 4 factors...
It turns back into a stem cell. It forgets the heart muscle cell.
And stops pumping blood, which is not helpful.
Definitely not the goal, no.
Yeah.
So that realization led to the next big idea. Partial rejuvenation.
Partial, meaning?
Meaning you don't go for the full reset. You try to apply the Yamanaka factors maybe for a shorter time or at a lower dose.
Yeah.
Just enough to clear out the epigenetic noise, maybe lengthen telomeres a bit, boost the mitochondria.
But without making the cell lose its identity. It stays a heart cell or a neuron or whatever it is.
Exactly. You're aiming to restore youthful function without wiping the slate clean. It's more like tuning up an engine than replacing it entirely.
And has that worked? I mean, in animals. The sources mention mice living 50% longer.
Yeah, those studies were pretty eye-opening. A 50% increase in lifespan is significant, even in mice.
It is, but we always hear it worked in mice. Does it translate? What makes researchers think this partial approach might work in something as complex as us?
Well, a really compelling piece of evidence came out in 2020. Dr. David Sinclair's group used this partial rejuvenation method.
Sinclair, yeah, big name in aging research.
Right. They used it to restore vision in mice. These mice had damaged optic nerves, either crushed or damaged from high pressure, like glaucoma.
And they used the Yamanaka factors.
The same factors, yes, but delivered in a way that caused partial rejuvenation. And the damaged nerve cells actually repaired themselves and regenerated. Function was restored.
Wow, so it wasn't just making new cells, it was fixing specialized adult cells in place.
That's the key takeaway. It showed these factors could potentially repair damage within a complex issue without just creating generic stem cells. That's a very different proposition.
That is a big deal. Okay, what about in human cells? Any progress there? The sources mentioned a 2021 study.
Yes, another important step. Scientists took human cells, I think fibroblasts again, with a measured biological age of about 60 years.
Okay, definitely mature cells.
And they exposed them to a partial rejuvenation protocol using these factors.
And the result?
After just 13 days. 13. Those 60-year-old cells were measured as having a biological age of 25.
13 days to roll back the clock, 35 years.
Biologically speaking, yes. According to those epigenetic markers, telomere length, mitochondrial function, they look like cells from a 25-year-old. And critically, they retained their identity as fibroblasts.
That's staggering. It really suggests age is fluid at the cellular level.
It really does seem to be malleable, yes.
So if the science is getting there, what's the holdup for, actual human therapies?
The biggest hurdle right now is delivery. We need a reliable, safe vector.
A vector, like a delivery system.
Exactly. A way to get these factors into the right cells inside the body in precisely the right amount for precisely the right duration to achieve that partial rejuvenation effect.
Without having to take cells out, treat them in a lab, and put them back in, which is cumbersome and expensive.
Right. We need an in vivo delivery system that's controllable and safe. That's the current bottleneck. Finding that vector is kind of the holy grail for the field right now.
Okay. So let's pull back a bit. We've redefined aging as communication breakdown. We've seen cells reset in dishes, partial rejuvenation in mice restoring vision, and human cells getting biologically younger. What does this actually mean for you, the listener? Why should you care beyond just living longer?
Well, it shifts the focus entirely. It's less about just lifespan and more about health span, living better.
Reducing disease risk, you mean?
Absolutely,
Yeah.
because the fundamental cellular communication pathways that these Yamanaka factors seem to be fixing, they're deeply implicated in major chronic diseases. The sources specifically mention things like the WNT signaling pathway. When WNT signaling goes wrong, gets mutated or misregulated, It's linked to a whole host of nasty stuff,
Like what?
like breast cancer, prostate cancer, colon cancer, pancreatic cancer, liver cancer, also diabetes, glioblastoma.
Mm.
The list goes on. These pathways are critical for normal cell function and development.
So fixing the underlying communication failure.
Could potentially reduce the risk of developing these diseases in the first place.
Yeah.
It's tackling the problem at a much more fundamental level than just treating symptoms after the disease appears. It's changing the environment that allows disease to thrive.
That reframes things completely. It's not anti-aging. It's like molecular level disease prevention.
You can certainly see it that way.
And it also links back to something you mentioned earlier, that incredible regenerative ability we seem to lose as we age, like newborns regrowing fingertips.
Right. That capacity exists. Babies and very young children have amazing regenerative potential. A lost fingertip can sometimes regrow, almost perfectly.
But that ability disappears around age 6 or 7. Why?
It seems to be an epigenetic shutdown. As we mature, the body seems to prioritize stability and maybe energy conservation over rapid growth and regeneration. Those youthful repair programs get silenced.
So the Yamanaka factors could potentially reawaken those programs.
That's the hope. If we can gently nudge the epigenome back towards a more useful state, maybe we can regain some of that lost regenerative capacity.
We already see hints of this with stem cell therapies, like for severe burns where they spray stem cells.
Exactly. Stem cells are powerful because they are regenerative. But imagine being able to coax your own existing cells to repair damage in place, using their latent youthful instructions. That could be even more powerful.
Yeah, much more targeted.
And that really changes the whole conversation about longevity, doesn't it? It's not about clinging to life at 100 when you're frail and your senses are failing. It's about having the energy, the eyesight, the hearing, the strength, the mental clarity to actually enjoy those later years.
To maintain purpose and quality of life.
Exactly. It's about changing the biological identity from old and declining to long-lived and functional. Healthy longevity. That's the real prize here.
Okay, so wrapping this up, the big takeaway seems to be that our cellular age isn't set in stone, it's dynamic.
Very dynamic.
And the control panel seems to be the epigenome, with these 4 specific Yamanaka factors acting like master switches.
That's what the science is strongly suggesting, yes. They hold incredible potential to influence that biological clock.
Which leaves us with a final thought, maybe something for you, the listener, to really chew on.
Go for it.
If aging, at its core, is this kind of communication breakdown, and if that breakdown is linked to so many major diseases we currently think of as just inevitable parts of getting older, and if we're learning how to potentially fix that breakdown, to rewind the cellular clock, what does that really mean for our whole concept of age-related disease?
Yeah. Is disease truly inevitable? Or is it perhaps often just a sign of repairable communication failure at the cellular level?
A pretty profound question to ponder.
Definitely something to think about until our next deep dive.